Assalamu alaikum. Welcome to my lecture. Welcome to my video. So, in this lecture today, we will discuss about the genomics and proteomics. We will discuss the introduction to genomics and proteomics for beginners. So, basically, what are these things and how these things are interrelated and why we required uh, to know to know these genomics and proteomics. So basically, these are the new field having the new vocabulary. For this, we need to recall our concept of the genetic flow of information. That basically the flow of information from DNA into the DNA towards the proteins. Now, what does that mean is our cells use DNA to create the RNA molecule. So essentially, our DNA molecules contain specific genes. These DNAs are basically the blueprints of life. These are the double-stranded molecule and they are the polymers of the deoxyribonucleotides. So as I already told you that our DNA molecules contain specific segments of the nucleotides जिनको हम genes कहते हैं. बेसिकली व्हाट दे डू, ये जो न्यूक्लियोटाइड्स होते हैं ये एक स्पेसिफिक कोड लैंग्वेज बनाते हैं, कोड लैंग्वेज ऑफ डीएनए इन द फॉर्म ऑफ न्यूक्लियोटाइड्स that that ultimately code for the different types of the proteins. Where do these proteins come from? They actually come from the information from the DNA, that is the information that is stored in the DNA. Now, if you want to make a copy of your own DNA, this is the replication. Now, if this code, this specific information, why am I saying the code or a code sequence? For example, if for a computer has a central processing unit जिसको हम CPU कहते हैं, उसकी एक अपनी लैंग्वेज होती है in the form of binary digits. जैसा कि मैंने आपको बताया कि जो डीएनए है दैट इज आल्सो द रिपोजिटरी ऑफ इंफॉर्मेशन. डीएनए में इंफॉर्मेशन स्टोर्ड होती है इन द फॉर्म ऑफ स्पेसिफिक कोड लैंग्वेज. तो मैं आपको सेंट्रल प्रोसेसिंग यूनिट का बता रही थी कि उसकी एक अपनी लैंग्वेज होती है, कोड लैंग्वेज इन द फॉर्म ऑफ बाइनरी डिजिट्स. दोस बाइनरी डिजिट्स आर दी 0101. ठीक है, 01 की फॉर्म में जो कोडिंग लैंग्वेज होती है वो इंस्ट्रक्ट करती है in order to perform the various functions of the computer. So same goes for DNA. DNA has having that particular code language in the form of a coding sequence of nucleotides. That particular sequence made up of the purines and pyrimidines. As let's say this DNA contains information in the form of A, G, C, T, T, G, C, T. ठीक है, दिस इज द स्पेसिफिक सीक्वेंस ऑफ द न्यूक्लियोटाइड. दिस इज द कोडिंग सीक्वेंस. अब यहाँ पर क्या होगा, दिस इंफॉर्मेशन जो कि डीएनए में प्रेजेंट है इसको आरएनए रीड करता है, दैट इज स्पेसिफिकली द एमआरएनए, एंड दिस प्रोसेस इज नोन एज द ट्रांसक्रिप्शन. ये जब एमआरएनए इस इंफॉर्मेशन को रीड करता है, it will read this particular information in its own language. For example, अगर इसकी लैंग्वेज में बात करें एमआरएनए में, तो देन दिस इंफॉर्मेशन विल बी लाइक ए जी सी यू यू जी सी again you, because you know that DNA is a double stranded molecule. DNA के अंदर थायमिन न्यूक्लियोटाइड होती है, थायमिन बेस होती है, but in RNA have uracil instead of thymine. So ये जो mRNA है ये DNA की लैंग्वेज को, इंफॉर्मेशन को अपनी लैंग्वेज में इसको रीड करता है. आफ्टर दैट इट विल ट्रांसलेट दैट इनटू द प्रोटीन्स. इसको रीड करके वो अलग अलग तरह के प्रोटीन्स बना देता है जिनके हमारी बॉडी के टिश्यूज में स्ट्रक्चर, उनके फंक्शन, उनकी रेगुलेशन में इन्वॉल्व होते हैं. यू नो दैट प्रोटीन्स आर बेसिकली द डाइवर्स ग्रुप, diverse, they have diverse function in our body. So this omics is basically the area of research. Now we have the area of research having the new vocabularies. For example, in this flow of information from DNA to mRNA and to protein, this is referred to as the central dogma. इसको हम कहते हैं central dogma of life, or you can say the central dogma of molecular biology. Now the new vocabulary for the DNA, the complete set of DNA is the genome. Genome referred to as a complete set of genome, all the chromosomes, genes, they comes under the genome. So for RNA, all total RNA content in the cell is the transcriptome. And then when it comes to proteins, all the proteins in the cell, that is referred to as proteomes. So in case of omics, the study of the research on genome, the study of the complete set of genome is the genomics. It will involve the complete study of the genes, genome in an organisms, that will involve their sequencing, that will involve analyzing their structure, their function, as well as their evolution. Now, basically, what happens? This genomics, it provides insights into the their genetic variation, the relationship between the genes and the disease. Now, this is the genomics from genome. In the classical genetics, now this from genome to transcriptome to proteome, this is the classical central dogma of molecular biology. Now if we extend this to metabolites, as the proteins have to metabolize, and these are the metabolites, the whole metabolic system is basically the metabolome. Now, if we talk about the modern central dogma of the molecular biology, then it also involves the metabolome. Metabolome and the metabolomics is basically the study of the metabolome. Metabolome, this हम क्या कहते हैं? Metabolome is basically the whole system. It is the whole metabolic system containing all the metabolic processes. That is the metabolome. It will involve the protein interactions of the protein with the different metabolites, sugars, amino acids, and other processes like glycolysis, Krebs cycles, and all these metabolic pathways, they are interrelated to each other. Therefore, the study of the whole metabolic system, the study of the metabolome is basically the metabolomics. Now, in the modern central dogma, this metabolomics, this metabolome is involved. Now, according to the classical central dogma, it is the flow of information that is from the genome to transcriptome to the proteomes. Now, if we talk about the modern central dogma of molecular biology, then it is the genome to transcriptome, then proteome to metabolome. So these terms, these research areas, they belongs to the, as in case of genomics, it belongs to the study of genomes. Transcriptomics, it belongs to the study of complete study of the transcriptome, having their RNA, their transcript profiles. Similarly, there for proteomics, the complete study of the proteomes, including their protein expression profiles, and metabolomics is the study of the whole metabolic processes, the whole metabolome. Now let's come towards the technical definition of the genomics. When it comes to the technical definition, then genomics is basically the study discipline of biology. Now, if we more specify it more specifically, it is the study discipline of the genetics, as it basically belongs to the study of the genes, study of the genomes, study of the gene, study of the genome. That basically applies the DNA sequencing methods and recombinant DNA technology, and also make use of the bioinformatic tools to sequence, assemble the huge amount of the data, and then analyze their structure and the function of the genome. Now, for each of the omics, each of the genomics as well as proteomics and transcriptomics, we require a certain setup. We require different types of equipments. We require different types of instruments for analyzing our data, analyzing our results. Now, actually, the purpose of this is actually we want to know that what's going on. Okay, basically, we want to understand their structure. We want to understand the structure of protein. We want to understand the function of the protein. ठीक है, सो इसके लिए हमें एक सर्टेन सेटअप रिक्वायर होता है. फॉर एग्जांपल, इन केस ऑफ genomics, we need a specific instrument, specific equipment, that is the DNA sequencer, that will basically use to sequence the genome. कि genome जो है, जीन क्या है, इसका specific sequence क्या है, sequence किस चीज का, that coding sequence of the nucleotide. Then this sequencer will give us the sequence data. फिर उस डेटा को हम जब प्रॉपरली एनालाइज करेंगे थ्रू द यूज ऑफ डिफरेंट सॉफ्टवेयर्स एंड टूल्स, and then we get the all the information regarding genome. So this comes under the category of the genomics. And now these genomics basically classified into four disciplines. That is the structural genome, into four categories. These are the structural genomics, functional genomics, and comparative genomics, and mutational genomics. When it comes to the structural genomics may we want to understand the structure of the gene products, as this is the data is a high throughput data, huge amount of data. So all these study their complete sequences, their complete profiles. This comes under the category of the structural genomics. Next is the functional genomics. Functional genomics basically the study of the genomics as well as transcriptomics. इसमें हम क्या करते हैं कि हमें जो जीन्स है उनके फंक्शंस को आइडेंटिफाई करते हैं, जो कि हमें हेल्पफुल होता है डिस्क्राइबिंग द जीन्स फंक्शन, के उस पर्टिकुलर जीन का क्या फंक्शन है, किस तरह के वो प्रोटीन्स बनाएगा, क्या क्या उसमें चेंजेस हैं. एंड देन वी वांट टू अंडरस्टैंड द जीन एंड प्रोटीन इंटरेक्शन. So, or if we analyze such things, if we analyze their interactions, then this comes under the category of the functional genomics. Now, if we want to manifest the genotype. Genotype is the genetic makeup, or the total genetic content is the genotype, and phenotype is the physical appearance of a trait. Many individuals have different physical characteristics, different physical attributes, जिनको हम traits कहते हैं. For example, if we talk about hair color, so different लोगों का जो hair color होता है, that is the different. कुछ लोगों के ब्लैक बाल होते हैं, कुछ के ब्राउन होते हैं. ठीक है, सो ऑल दिस बेसिकली डिपेंड्स अपॉन द जीनोटाइप, के उसकी क्या जीनोटाइप है, क्या उसका जेनेटिक कंटेंट है जो कि उसके ये फिनोटिपिक फीचर्स को डिस्क्राइब कर रहा है. सो इस तरह की स्टडी को हम क्या कहेंगे, दिस इज द फंक्शनल जीनोमिक्स. फॉर एग्जांपल, आई कलर हो गया. ठीक है, कुछ लोगों का आई कलर ब्राउन होता है, कुछ की आईज ग्रीन होती हैं, कुछ की ब्लू होती हैं. ठीक है, तो फिनोटाइप इज बेसिकली द मैनिफेस्टेशन ऑफ द genotype. All these For all these things, for all these analysis, we need to have certain technologies. In this case, we have for example, DNA microarrays, and in order to know the expression and the splicing events, if we want to know the splicing events, these are the basically transcriptional modifications. This comes under the category of the functional genomics. In case of comparative genomics में हम क्या करते हैं, जो different species का genome है उस इनको आपस में कंपेयर करते हैं. कंपेयर क्यों करते हैं, जिससे हमें पता चल जाता है कि डिफरेंट स्पीशीज के दरमियान में जो सीक्वेंसेस है उनकी कितनी सिमिलैरिटी है, या कितनी वो डिफर कर रही हैं. ठीक है, सो अगर जहाँ पर हमने स्टडी करना है एवोल्यूशनरी स्टडीज को, या एवोल्यूशनरी रिलेशनशिप्स को हमने स्टडी करना है, तब हम किस पे जाएंगे, देन वी विल गो फॉर कंपैरेटिव जीनोमिक्स. सो कंपैरेटिव जीनोमिक्स में बेसिकली क्या होता है, इट विल compare the genome sequence between the different species, and it will help us to better understand the evolutionary relationships among different species. And then next is the mutational genomics. Mutational genomics, as यहाँ पर we need to study the details about the mutations. We know there are certain types of mutations के जो nucleotide में change आ जाता है. Certain types of mutation include insertions, deletions, SNPs, and the frame shift mutations. The thing is that whenever we want to analyze the mutational changes, mutations in DNA और genome of an individual, तो इस चीज की स्टडी को हम mutational genomics कहेंगे. You know that the RNA, DNA, they have a coding as well as non-coding sequences. In case of messenger RNA, they have introns and exons. Introns are basically the non-coding, जो कि किसी भी प्रोटीन को कोड नहीं करते हैं, एंड दी एक्सॉन्स आर द कोडिंग सीक्वेंसेस जो कि डिफरेंट प्रोटीन्स को कोड करते हैं. अगर इन सीक्वेंसेस में कहीं म्यूटेशंस आ जाती हैं तो इसकी वजह से जो इनका फाइनल प्रोडक्ट होता है, जो प्रोटीन्स है या जो भी इनका फाइनल प्रोडक्ट होता है, वो ठीक से नहीं बनता. सो इस चीज की डिटेल में, इस चीज की स्टडी को हम कहते हैं दैट इज द म्यूटेशनल जीनोमिक, स्टडी ऑफ द जीनोम इन टर्म्स ऑफ mutations in the DNA of a individual. So all these are the genomics. When we have to describe proteomics, we have to encounter the transcriptomics. You know, transcriptomics basically belongs to the transcriptome. Transcriptome is the all the sets of the RNA molecules. So the research, the study discipline in which involves the study of the sets of the all RNA molecules inside the cell. We have like messenger RNA, transfer RNA, ribosomal RNA, and then nuclear RNAs. There are different types of RNA. अब messenger RNA basically क्या करता है? Messenger RNA that contains the sequences of the ribonucleotide. What I told you earlier कि ये जो ribonucleotides होते हैं basically ये DNA की जो information को read करता है in its own न्यूक्लियोटाइड इन इट्स ओन लैंग्वेज. ये जो राइबोन्यूक्लियोटाइड होते हैं ये कोड करते हैं स्पेसिफिक अमाइनो एसिड सीक्वेंसेस को. वो जब अमाइनो एसिड्स एक स्पेसिफिक ऑर्डर में ऑर्गेनाइज होते हैं जो कि इनको इंस्ट्रक्ट कर रहा होता है डीएनए, देन अल्टीमेटली दे फॉर्म प्रोटीन्स. सो दीज आर द mRNA contains basically the sequences of the ribonucleotides that code for the specific amino acid sequences for the synthesis of proteins. Next, this ribosomal RNA basically क्या करता है? It forms the part of the structure of the ribosomes. Ribosomes actually these are the sites of the protein synthesis. अब जो transfer RNA है ये क्या करता है? These molecules carry the amino acids to the ribosomes because these are the ribosomes that are actually the sites of the protein synthesis. जो proteins है वो यहाँ पर बनते हैं. तो ट्रांसफर आरएनए जो mRNA की कोडिंग के हिसाब से अमाइनो एसिड्स जिनको mRNA कोड कर रहा होता है, एक स्पेसिफिक कोड कर रहा होता है, वो mRNA वो अमाइनो एसिड्स को ये ट्रांसफर करते हैं टुवर्ड्स द साइट ऑफ प्रोटीन सिंथेसिस. So we need some techniques like microarray technique that will provide us with the messenger RNA profile, and after that we need to have deep investigation. We need to have deep study about the transcriptional activity, that is the transcriptome. So the study of the complete set of the all the RNA molecules having the RNA profiles, the analysis of those profiles, interpretation of the data, and high throughput data. This comes under the category of the transcriptomics. When it comes to the proteomics is basically the large scale study of the proteins. प्रोटीन्स की स्टडी में हम क्या करते हैं? इनके डायनेमिक्स को स्टडी करते हैं, इनके स्ट्रक्चर को, फंक्शन को, दैट बेसिकली रेफर टू एज फंक्शनल प्रोटियोमिक्स. वी नीड टू एनालाइज देयर एक्सप्रेशन प्रोफाइल्स फॉर द सेल्स, टिश्यूज और ऑर्गेनिजम्स. अब नाउ प्रोटियोमिक्स बेसिकली क्या करता है? ये जो प्रोटीन बेसिकली ये क्या करता है? वी नीड टू compared the expression profile of the proteins कि डिजीज इंडिविजुअल है और एक नॉर्मल हेल्दी कंट्रोल है, हेल्दी इंडिविजुअल है, इन दोनों की जब प्रोटीन प्रोफाइल को कंपेयर किया जाता है देन वी नीड टू वी बेसिकली नो अबाउट देयर एक्सप्रेशन के डिजीज में का जो एक्सप्रेशन होगा वो नॉर्मल से डिफरेंट होगा, इधर इट इज इट गोना हाई एक्सप्रेशन और अ लो एक्सप्रेशन. So the information regarding the protein expression profile of the cells, tissues, and organisms that basically provides understanding of the biological processes and the disease mechanisms uh, for the particular disease. So proteomics is the large scale study of the protein complement of an organism, including their structure, structural, and function. That is the functional proteomics. तो इसमें क्या है, जो ये कंप्लीट सेट ऑफ प्रोटीन है जिसने बेसिकली क्या करना है, एक्सप्रेस, दैट इज एक्सप्रेस्ड, फिर बाद में क्या होगा, दैट विल बी मॉडिफाइड फॉलोइंग द एक्सप्रेशन बाय द, जो कि इसको इंस्ट्रक्ट कौन कर रहा है, जो कंप्लीट जो जीनोम है. ठीक है. So this proteomics is basically the scientific discipline that will involve the large scale study of the protein. ठीक है. It will basically provides the understanding of the genome sequences and then their cellular behavior. कि basically these are the ultimately these are the proteins जिन्होंने अपने cell में, body में different roles perform करने हैं. Proteins have diverse roles in our body, and this proteome is basically the entire set of proteins that are produced by the different cell types. These proteins in all the enzymes are basically proteins in nature and they act as catalyst. Exceptions are always there, you know, that these the ribozymes are not entirely these are proteins. ठीक है. So basically जो enzymes होते हैं normally these are the globular proteins and they act as catalyst. इसी तरह proteins are also act as a regulatory molecules. For example जो hormones होते हैं जो कि regulation में involve होते हैं. ठीक है. These are also the proteins. Similarly proteins also act as transport proteins. For example hemoglobin जो कि help करता है in the transport of oxygen to various tissues. Similarly जो हमारा defense system है, हमारा immune system है, that also have proteins in the form of antibodies that defend against the foreign particles. So in short these proteins have a diverse functions in our body. ठीक है. So these proteomes जो है they basically can be studied using the knowledge of genomes. So that's why all these things are basically interrelated. For this we need to have instruments like 2D gel electrophoresis that will give us with the protein profile, and in proteomics we need to identify, we need to characterize the proteins. It will involve the protein-protein interactions or interactions of proteins with other molecules and identify the disease biomarkers. This will provide the understanding of the disease mechanisms as well. So the complete study of the proteome is basically the proteomics. Now, basically, what happens? All these things are interrelated. These are interrelated with the bioinformatic tools. You know that the bioinformatics is basically the established resource for the molecular biology research. ठीक है. And basically जितने भी सॉफ्टवेयर्स हैं, tools हैं जो हम डेटा को स्टोर करने में, एनालाइज करने में यूज करते हैं, that all those they belongs to the bioinformatics. Okay, now there are huge number of these sequences that are stored in the genetic databases that are basically form of the bioinformatic tools. We need to sequence all the genetic databases from the variety of the organisms, including human genomes. So basically what happens ke this bioinformatics is now the vital resource for the molecular biology research. All these are interrelated to the bioinformatics, this genomics and the proteomics as well. In proteomics basically क्या होता है कि जो proteomics है, that characterization of the genome functioning on the protein level. What does that mean? कि these proteomes जो हैं, they can be studied using the knowledge of the genomes. First of all, we have knowledge of the genomes. जब तक हमारे पास genome की information नहीं होगी, how can we study the proteome? ठीक है, so these proteomes can be studied using the knowledge of genomes, because genes code for the mRNAs, and then this mRNAs क्या करते हैं, they basically encode the proteins, क्योंकि mRNA में वो sequences होते हैं जो कि proteins बनाते हैं. The study of the इसी तरह जो इन प्रोटियोम्स के फंक्शन की स्टडी को हम कहते हैं, दिस इज द फंक्शनल प्रोटियोमिक्स. अब नाउ बेसिकली व्हाट हैपेंस कि जो ये रिसर्च है, एज द ओमिक्स इज द रिसर्च एरिया, सो इन केस ऑफ प्रोटियोमिक्स बेसिकली कॉम्प्लीमेंट्स द जीनोमिक्स. और ये यूजफुल कब होता है, when the scientists want to test their hypothesis that were basically based on the genes. ठीक है, तो जब इनका हाइपोथेसिस जीन्स पर बेस कर रहा होता है, तो उससे फिर वो इस रिसर्च को फुलफिल करने के लिए प्रोटियोमिक्स की स्टडी करते हैं. नाउ जीनोम्स एंड प्रोटियोम्स ऑफ द पेशेंट्स सफरिंग फ्रॉम द स्पेसिफिक डिजीज, इफ वी वांट टू स्टडी द genetics of the specific disease, we have to go for their genomics and proteomics. Therefore, if we want to study the genetic basis of particular disease, then we have to study the complete genomes, complete genomics, complete proteomes. We have to study their proteomics of the patients that are suffering from the specific disease. So these are being studied to understand the genetic basis, to elucidate their genetic basis of particular disease. Now, जो proteins में most important prominent disease है जिसको study किया जा रहा है, जिस पर research की जा रही है, that is basically the proteomic approach is the in the cancer research. ठीक है, these proteomic approaches, these are being used to improve the screening and the early detection of the uh, cancer, and this is basically achieved by identifying the proteins whose expression is affected by the disease process. Now this proteomics will give us the expression profile of the of the proteins. ठीक है, then we need to compare those profile with the normal healthy control healthy individual, and after that we will analyze their results. When we compare their profile, कि हम disease person और normal person की जब profile को compare करते हैं, there is a change of expression. ठीक है, जब expression या तो disease person में high होता है या low होता है, तो यहाँ से हम इसकी जो genetic basis है उसको basically study करते हैं. So all these genomics and proteomics, these are the interrelated. In genomics, we need to study the complete structure and functional aspects of the genome. So this was all about the introduction to genomics and proteomics, and this is for the beginners. If you like this video, then don't forget to subscribe my channel. Thank you very much, and best wishes for all. Thank you.